Although humans aren't predators like sharks, we too have very efficient teeth, which enable us to eat large food sources like this apple. We call animals that feed on large food particles macrophagous feeders, and this includes animals like sharks and ourselves. But macrophagous feeders don't necessarily have to have teeth to be able to feed on large food sources. And in this program, we'll see a number of examples of macrophagous feeders which don't possess proper teeth at all. When we come to classify animals according to the way in which they feed, we find a number of examples of closely related animals that feed using entirely different sorts of mechanisms. Conversely, we find a number of unrelated animals which use feeding mechanisms that operate on similar principles. The nature of the food available can be as important as the class to which the animal belongs in determining the type of feeding mechanism used.
The ragworm's a polychaete, a common seashore annelid. It's a carnivore and has a muscular pharynx with large horny jaws. The pharynx is shot out through the mouth, opening the jaws, which can seize the prey. The pharynx then retracts, and the prey is gripped by the jaws and swallowed. In this section, you can see the position of the retracted pharynx and of the jaws. The pharynx is controlled by the opposing protractor and retractor muscles, and coelomic pressure also helps. It's unusual to find a phylum in which all the animals feed in one way, but this is the case with the Cnidaria. Though lacking jaws, they're carnivores. Their tentacles carry cnidoblasts, which contain stinging structures called nematocysts. They're so efficient that this hydra catches a water flea almost as large as itself. Under a microscope, you can see the nematocysts discharging. The tiny threads pierce the prey and inject a paralyzing poison. But what stimulates this discharge? If the anemone is touched with a clean glass rod, given a mechanical stimulus, there's very little feeding response, and the nematocysts do not discharge. Given a chemical stimulus, a little juice from a crushed prey animal in this case, again there's no feeding response, and nematocysts do not discharge. But if a mechanical and a chemical stimulus are given together, there is a feeding response. The nematocysts discharge, and the anemone's tentacles seize the piece of the prey and move it towards the mouth, which is in the center of the ring of tentacles. Under phase contrast, the coiled thread within an isolated nematocyst is clearly visible. During discharge, the thread is turned inside out extremely quickly. The common starfish, Asterias rubens, is also a carnivore. It feeds on shellfish, which are often much too large to fit within its stomach, as well as being surrounded by a hard, indigestible shell. So, the starfish wraps its arms around its prey, a whelk in this case, holding tight with its tube feet. And then it turns its stomach inside out. The stomach wall secretes enzymes. These digest the body tissues of the whelk, and the resulting soup is then absorbed by the stomach of the starfish. Given long enough, there's nothing left but an empty shell. But if the starfish is lifted off the whelk, the stomach's briefly visible. The common mussel, Mytilus, poses the starfish an additional problem, as it has to get inside two shells. It does this by wrapping its arms around the mussel, slowly pulling the two shells apart, and then inserts its stomach in the gap. These characteristic marks show that a limpet once lived here. These are very common marine herbivores. Some gastropod mollusks, like limpets, graze on seaweeds and algae, rasping away at food with their radula. Under a microscope, the radula, a ribbon-like structure, can be seen to have rows of fine teeth. The periwinkle feeds in the same way, rasping food into small particles, which is then swallowed. But some gastropods have a very different radula. Whelks, for instance, are predators. With their powerful radula, they rasp away the shell of their prey. Scanning electron microscopes shows the detail of their radula. As the teeth at the front of the radula are worn out, teeth further back move up to replace them. Conus is a fearsome predator. A few radula teeth have become enlarged, and these form pointed darts. They're fired into the prey and inject a deadly venom. All the examples we've seen so far have been of macrophagous feeders. But this polychaete worm, called Pomatoceros, though closely related to Nereus, is not macrophagous. It filters out tiny food particles that are suspended in the water in which it lives, using a crown of tentacles. Captured food particles are passed down along the tentacles to its mouth. Animals that feed on tiny particles are called microphagous. The barnacle's another filter feeder. Adult barnacles are sedentary, permanently attached to their substrate, and the limbs are modified for feeding. They're fringed with setae and sweep the water rhythmically, collecting food particles. Some of the most sophisticated examples of filter feeding are found amongst the lamellibranch mollusks. The mussel opens its two shells slightly, allowing a current of seawater to pass through. Outwardly, the animal doesn't show a lot of activity associated with feeding. But inside, there are gills covered with cilia. Their wave-like beating creates a current of water over the gill's surface. This current carries food particles with it, and as the current passes over the gills, the food is trapped by interlacing cilia. Mucus binds the particles into food strings, which are passed along the edge of the gill to the mouth. Before entering the mouth, the particles are sorted, and unwanted material is carried away on a water current. Here you can see the rows of cilia on a gill, transporting food particles to the edge of the gill and then along to the mouth. And in slow motion, here's the action of a single cilium. The real intricacy of the gill can only be seen with a scanning electron microscope. The cilia can trap particles as small as 1 micron across. Nearly all the filter feeders are invertebrates, but there's one exception, a basking shark. With its characteristic gape, it sieves plankton from the sea in which it lives. A completely different type of feeding occurs in those animals that feed on fluid or soft tissues. The platyhelminthes include many free-living predators. Their primitive gut has only one opening at the end of a muscular pharynx. A vertical section through a planarian shows the pharynx within the body. A freshwater crustacean falls prey to a planarian. When feeding, the pharynx is shot out. It produces digestive enzymes that enable it to pierce the body of its prey, and once inside, to dissolve the body tissues. These can then be sucked up into the digestive system of the flatworm. The pharynx interesting exception, a vertebrate, a vampire bat. Their sharp incisors painlessly pierce the skin of their host. Blood pours out, and this is sucked up and swallowed.
We've seen a number of examples of macrophagous feeders, animals which feed on large particles. We've also seen microphagous feeders, animals like bivalve mollusks, which filter small suspended particles from solution. Now we're going to look at an animal which feeds in an entirely different way. And to see it, I'm going to have to dissect this rat. I'm going to make a ventral incision along the abdomen. And I've now exposed the alimentary canal. We can see a number of interesting points in the animal's body cavity. We can see the liver. You can see a lot of fatty tissue here. And down underneath this fatty tissue, I can see the animal's stomach. The stomach is attached to the first part of the small intestine, which is the duodenum, which is down here. And the duodenum runs down into the rest of the small intestine, which is the coiled structures that you see along here. You can see that the small intestine is still moving slightly. These are the peristaltic webs because the animal has just been killed. Now, the animal that we're going to look at lives inside the intestine of the rat. So to see it, I'm going to have to remove the intestine. I'm going to snip the junction between the stomach here and the small intestine, which is along here. And now I'm going to strip out the small intestine. And as I pull it away, there's a lot of fat and mesentery, which I've got to be careful not to tear. It's quite a long intestine. And I'm going to cut it at the junction of the small intestine and the cecum. And now I'm going to flush the intestine out using a Ringer solution, a solution which is the same composition as the body fluids of the rat. Now I'll push the contents of the stomach down. I'm squeezing them down, squeezing them on down. And you can see how the intestine is filling up like a tube full of liquid, which in fact is what it is. And here we have the animal which we want to look at, tapeworms. And this is the rat tapeworm, Hymenlepis diminuta. And you can see how it's mixed up with a lot of the feces, but you can also see that we've got a number of tapeworms here. We've probably got about 7 or 8. And this rat was infected with tapeworm larvae about 2 months ago. And if I leave the dish alone, you can see how the tapeworms move around and wriggle on their own. If we look at this prepared section under the microscope, we can see at the anterior end, called the scolex, that there is no mouth. And as we go down the body of the tapeworm, we see that there's no sign of a gut. The tapeworm is segmented. The segments are called proglottids. And as we go further down towards the posterior end of the tapeworms, we see large proglottids containing eggs. So if Hymenlepis doesn't possess a mouth or a gut, how does it obtain its food? Remember that the tapeworm lives in the rat's intestine, and the intestine of the rat contains a lot of digested and partially digested food. So there will be amino acids, sugars, and fatty acids floating around in the lumen of the intestine, next to the external surface of the tapeworm. But how can the tapeworm utilize these nutrients? A clue comes if we look at a scanning electron micrograph of the surface of the tapeworm. This tegument is not an impermeable structure. In fact, it has similarities with the inner surface of mammalian small intestine.
Of our science faculty, and it's for the course Biology, Form and Function. The textbook image of the digestive system portrays nothing of its highly motile and dynamic nature. For our program, we use endoscopy, barium meal, and the gamma camera to show this.